Okay, this is the third of our online presentations, and it is part 2 of the organisation of the respiratory system, or the components of the respiratory system, and focuses on the microscopic regions of the respiratory system that are involved in gas exchange, principally as we introduced the alveoli. Now, I left the last online presentation with this diagram here, taken from Widmeyer. So just as a recap, the last online presentation, OL2, discussed the components or the organisation of the respiratory system, focusing on the conducting zone. We spoke about the nasal cavity, the pharynx, the larynx, the trachea, the bronchi, which have the cartilage, and the bronchioles, which don't have cartilage. They're the conducting zone because their only job is to deliver air from the atmosphere to the lungs, and vice versa, to deliver air from the lungs back to the atmosphere. There is no gas exchange that occurs here. And because there's no gas exchange, it's commonly called the dead space. That is, the volume of these structures are called the dead space, and it has an average volume in a typical adult male, which is the archetypal physiological person, of about 150 mils. As I said, it's called the dead space because there's no gas exchange that occurs. Now, the importance of the dead space will become very obvious when we discuss this in the live lectures throughout the respiratory module. What we're going to talk about today in this presentation is the respiratory zone, which is where gas exchange does occur. And we spoke about briefly the respiratory bronchioles, which are the last components of the bronchial tree, the alveolar ducts, which basically, if you think of the alveoli, they're kind of like the stalks. If you think of the alveoli as a bunch of grapes, the alveolar ducts are the little stalks, and the alveolar themselves, which form the vast majority of the respiratory system. As I said, about 99.99%, I kind of made that up, but nearly all of the gas exchange that occurs in the respiratory system occurs in these alveoli. So let's look at them in much more detail. Now, if we look at this diagram, well, basically, before we look at the diagram, we've got the alveoli. We will see in the diagram in a moment that the alveoli contain 3 different types of cells. Firstly, we have the simple squamous epithelial cells, which are commonly called the type 1 cells. Now, this squamous epithelial, the squamous means that they have irregular shapes, or different types of shapes, and epithelial again reminds us that it's the surface of the body. And these are important because they provide the wall of the alveoli. As we'll see in the next overhead, they're unusually thin compared to most other cells in the body, and that optimizes their role in gas exchange. The second type of cell we have are the macrophages. Now, these are immune cells which travel around the alveoli that we'll see in a moment, combating the pathogens that may have escaped all the other protective mechanisms we spoke about in Online 2. So if they get to the alveoli, these macrophages hopefully will do the job and remove this debris and microbes from the inner surface of the alveoli, thus protecting them again from infection or inflammation. Now, obviously, if the alveoli did become infected or inflamed, it would thicken the wall of the alveoli, and that's going to compromise gas exchange. You don't want gas exchange to occur across a thick wall. In fact, it probably won't be able to occur. And the third type of cells we'll see in the next overhead are the surfactant secreting cells, or the type 2 cells. Surfactants is a watery substance containing phospholipids, or lipoproteins, which are kind of just like a detergent. Now, why is this important? Well, the liquid of the surfactant produced by these type 2 cells lines the internal surface of the alveoli with a very thin coat of liquid. Now, this is important because gas exchange can't occur effectively across a dry membrane. So the problem with water, though, is that when it has a really strong cohesive force, that is, the water molecules really want to stick together. Now, given that alveoli inflate and deflate rather like a balloon when we breathe, if the walls of the alveoli had a single layer of water lining them, and that these water molecules are very strongly cohesing together, stretching these water molecules in order to inflate the alveoli would be nearly impossible. It would be very difficult, and it would make the work of breathing much higher than it currently is. So in order to overcome that, we need the liquid to optimize gas exchange. We don't want the water bonds sticking, water molecules bonding together very firmly, so we need to produce something which weakens the bonds between the water. And this is what surfactant does. Now, the way I like to think of it this way, is if you imagine two sheets of glass sitting on top of each other. When dry, easy to separate. When they're wet with water, they're virtually impossible to separate. So if you add detergent to the water and then put them on top of each other, you can now separate those two plates of glass much more easily. And that's because the surfactant or detergent is weakening the cohesive bonding of the water molecules. Now let's look at this in situ. So in fact, if we look at a diagram taken from Widmeyer, figure 13.4, we now see the alveoli. So, and we, in the bottom left-hand part of the diagram, we've got the type 1 cells indicated in pink, these squamous, irregular, epithelial-shaped cells providing the wall of the alveoli. We have the macrophages indicated in blue. In fact, these are the macrophages here, and so they're travelling around the inner wall of the alveoli, cleaning up any debris and particulate matter. And then we have the surfactant secreting cells, secreting that detergent-like substance that I just spoke about. And they're the three major cell types of the alveoli. Now, if we go back to the alveoli and look at them, think of them more generally, we see that a typical human adult has about 300 million alveoli, each less than a micrometer thick. Now, the balloon shape of these alveoli, or the spherical shape of these alveoli, mean that we have an maximizing of the surface area. The large number of them, the 300 million or so that we have, means that within the lungs we've got a surface area collectively of about 70 meters squared. Now, if we consider gas exchange, or any exchange for that matter, across a wall, that you require a thin barrier and a large surface area. And so the alveoli fit that brief. So over 70 meters squared, and think of that as about half the size of a tennis court, of a membrane with a thickness about 1/1000th of a millimeter, 1 micrometer, surrounded by these capillaries, about 1,000 kilometers of capillaries, you can see that this is a fantastic adaptation to maximize gas exchange. So lots of spherical shaped alveoli, very thin walls, surrounded by lots of capillaries providing blood, great respiration or gas exchange in the lungs. So, as alluded to just then, and you can see here from this diagram, each alveolus is surrounded by a network of capillaries into which and from which gas exchange with the air that we breathe and blood occurs. We also see that the alveoli are surrounded by these elastic fibers. So you can see them in purple surrounding each of the alveoli. These elastic fibers obviously impart an elasticity to the lungs, thus allowing the alveoli to increase and decrease their volume so that air can move into them and out of them much more easily or appropriately. So we've got our alveoli, spherical shape, lots of them, surrounded by capillaries, lots of blood flow. Elastic fibers allow them to inflate and deflate rather like a balloon, important for inspiration and expiration. Thin walls, you can see very thin walls, less than a micrometer thick, so it's going to optimize gas exchange, oxygen moving from the alveoli to the blood, carbon dioxide principally from the blood back into the alveoli, which is exactly what we want to occur in normal respiration. This is an interesting diagram taken from Widmeyer figure 13.4, and it actually shows a more actual representation of a cross-section of alveoli. So there's a little bit of lung which has been sliced, and this is what we're looking at the top part of the diagram. So these would all be the alveoli here. Now, what do we learn from that? Well, clearly, you can see that even though the alveoli are the dead end of the respiratory system, they're the last bit which the air travels, then they're all interconnected via these pores or holes. So if we actually toggle back to the last diagram, you can see these holes, which are basically interconnections between successive alveoli. So if we look at them more clearly in this histological representation, the alveoli are all interconnected. And note that they're not all the same shape or size. So there are some bigger alveoli and smaller alveoli, and we'll talk about the significance of that in one of the live lectures. And finally, we see here that in fact, a bit more clearly, if we look at the top part, and I apologise, this diagram's not taken from Widmeyer textbook. I actually took it from another textbook called Saladin. It doesn't matter, you don't need to rush out and get the textbook. You can see the diagram here. Again, we see normal lung structure, and again we see what I just spoke about in the last overhead. Lots of alveoli, different shapes and sizes, all interconnected, very thin walls which make up lung tissue. Now, as I said, that's really important because it optimizes gas exchange. Exactly what we need. We want quick, rapid, maximal gas exchange. The problem is, if you've got, and we spoke in online presentation number 2 about all the protective mechanisms that the respiratory system, the conducting zone and the respiratory zone has to protect these very delicate structures. Now you can see that if in fact you got an infection or inflammation, the walls, like any infection or inflammation you have anywhere in the body, the walls thicken as a consequence, and you might even get this fluid build up around them. And now if this happens in the alveoli, obviously thicker walls and more fluid means that you're going to effectively reduce, if not abolish, appropriate gas exchange. Now this happens in diseases such as pneumonia, which is why pneumonia is often associated with lack of oxygen and excess carbon dioxide, which can ultimately kill the person. The other thing we see is that if this infection is chronic, that is constant, the infection and inflammation starts to break down the walls, and obviously we have a situation where somebody's had a chronic infection. The walls of the alveoli are no longer normal, they're broken down, and obviously that's going to reduce the surface area and reduce the opportunity for gas exchange. And a disease called emphysema, chronic emphysema, breaking down of the alveolar walls, massively reduce gas exchange, and again these people are symptomatic of having low levels of oxygen and high levels of carbon dioxide, which, as I mentioned previously in an online number 2, is toxic to the body and can obviously not be a good thing. Now in conclusion, I want to talk about ventilation and perfusion mismatch. What's that all mean? Well, basically ventilation, or V, refers to the air moving into the alveoli. We discussed that definition in online 1. Perfusion, known by the letter Q, or indicated by the letter Q, represents the blood flow via the capillaries to the alveoli. Now normally this needs to be matched in order to optimize respirational gas exchange. Now this is better understood if you see what happens when they're not matched. And this is an adaptation that we have. If we have a situation where the airflow to part of the lung, and let's look at the left-hand side of this diagram, which I took from Widmeyer, figure 13.24, or a diagram like it. If we have an area of the lung that has decreased airflow, so if this airflow is compromised due to damage, then obviously less air, fresh air, travels to those alveoli, which causes a reduction in the amount of oxygen and an increase in the amount of carbon dioxide in the alveoli, and hence the blood vessels travelling to them. This will cause a constriction, or the body responds by constricting the pulmonary vessels or the capillaries that are supplying those damaged alveoli, which means that we get decreased blood flow. So we again match the ventilation and perfusion. So decreased airflow to the alveoli because part of the alveoli are damaged. This causes a change in the amount of oxygen and carbon dioxide in them. The capillaries respond by constricting, reducing blood flow, and we now match with the airflow again. I mean, when you think of it, why would you send fresh blood to an area of the lung that is not working as it should? And vice versa, although this is obviously not as common in the body. Now, the opposite also happens. For example, if we've got damage to the capillaries, so if you've got a vascular problem where we have a decreased, and we'll look at the right-hand side because it's much more common, if we get a decreased blood flow to the alveoli, or to part of the lungs, obviously we're going to again compromise gas exchange. So if we can't send blood to the lungs, we can't deliver excess carbon dioxide to them to get rid of it from the body. And the alveoli respond, or the bronchioles respond to that by constricting and decreasing airflow to that part of the lung that's got a damaged blood flow, matching it again. Again, why would you bother sending air to a part of the lung that doesn't have appropriate blood flow or blood supply to and from it? So again, we tend to match ventilation and perfusion to maximize gas exchange. Now, how is this relevant to us? Well, I guess it's relevant because it means that we're always trying to maximize gas exchange in situations that you might have little problems with the lungs or little problems with the blood vessels that are supplying the lungs, but also in the normal lung. There's change in ventilation perfusion ratios. For example, if you're standing up, because the lung's positioned, if you think of it, the lung's positioned vertically around the heart, when standing up, the region of the lung above the heart is going to receive slightly less blood flow, because it's pumping against gravity, than the region below the heart. So this means that the VQ, or ventilation perfusion ratio of the lungs above the heart, is slightly higher. That is, more airflow, less blood flow, than the region below the lungs, below the heart, which is called the base of the lungs. So this ventilation perfusion matching is important in disease, but also important in the normal lung. It has some role. I wouldn't say it's important. It has some role in the normal lung, and we'll talk about the relevance of this in a bit more detail, probably in about live lecture number 3 from memory.